Hello everyone. Today we will discuss about amino acid metabolism. Amino acids are the building blocks of proteins. So proteins are linear polymers of L-alpha amino acids. Let's see some terms in amino acid metabolism. First one is amino acid pool. Amino acid pool consists of body's total free amino acids and it is about 100 grams. If a cell takes up as much as amino acid as it loses, it is in a state of dynamic equilibrium. If the loss is greater, the cell wastes. And if the gain is greater, the cell grows. Coming to the contributors of this amino acid pool, first one is digestion of dietary proteins, that is about 70 to 100 grams per day. Then synthesis of non-essential amino acids and degradation of old body proteins, that is about 300 to 400 gram per day. These are the sources. These proteins cannot be stored in the body. So proteins are catabolized in liver about 35 to 55 gram per day in the form of mostly urea. Then this urea, it is excreted through urine. So how these are utilized? There will be synthesis of new proteins, that is about 300 to 400 grams per day. Then synthesis of non-protein nitrogenous substances like porphyrins, purines, pyrimidines, creatinine, niacin, thyroid hormone, that is about 30 gram per day. This is amino acid pool. Next one is nitrogen balance. Nitrogen balance is defined as a balance between amount of nitrogen intake, that is in the form of dietary protein, and the amount of nitrogen loss or excretion in the form of urea, uric acid, creatinine and small amount of amino acids by an individual. So nitrogen balance is a balance between amount of nitrogen intake and the amount of nitrogen loss or excreted. When intake is greater than loss, it is termed as positive nitrogen balance. And it is seen in growing children, pregnancy and in recovery from illness. Hormones which lead to positive nitrogen balance include growth hormone, insulin and androgens. When intake is less than loss, it is termed as negative nitrogen balance and it is seen in acute or chronic illness, protein deficiency and in starvation. The hormones which lead to negative nitrogen balance include T4 and corticosteroids. Coming to amino acid metabolism, these amino acids constitute amino group and carboxylic group. And amino acid metabolism involves two steps. First one is removal of amino group and the second step is pathways or metabolism or fate of this carbon skeleton. In the metabolism of amino group, the first process is transamination. In transamination, there will be transfer of alpha amino group from one amino acid to a keto acid and form other new amino acid and a keto acid. And it is a reversible process. For example, aspartate plus alpha ketoglutarate with the enzyme SGOT, here PLP is required, and it gives oxaloacetate and glutamate. Here, aspartate is the donor amino acid and alpha ketoglutarate is the recipient keto acid. And the new amino acid formed here is glutamate. That is transamination. The second process is deamination. It is the process by which alpha amino group is removed as ammonia. Two types of deamination are there: oxidative deamination and non-oxidative deamination. In oxidative deamination, there will be coupled reactions, that is deamination and oxidation. For example, from this glutamate, ammonium is liberated and form alpha ketoglutarate. Here the enzyme is glutamate dehydrogenase. This ammonia is toxic. It will reach the liver and is converted to urea. Ammonia is transported in the blood in the form of amide or amino group. So next step is formation of glutamine. Here ammonia combines with glutamate and form glutamine in the presence of enzyme glutamine synthase. So ammonia transportation in blood is in the form of glutamine. Later, it is converted to glutamate and ammonium by the enzyme glutaminase. This ammonium then enters liver and form urea, which is less toxic. Then this urea, it is excreted through urine. This is the metabolism of amino group. Coming to the fate of this carbon skeleton, 4 fates are there. First one, oxidation by TCA cycle to provide energy. Second one, synthesis of non-essential amino acids. Third one, synthesis of lipids. And fourth one is synthesis of glucose. Carbon skeleton of 20 amino acids are converted to the intermediates of TCA cycle, either pyruvate or acetyl-CoA or acetoacetyl-CoA, alpha-ketoglutarate, succinyl-CoA, fumarate or oxaloacetate. There will be glucogenic amino acids, ketogenic amino acids and both gluco and ketogenic amino acids. These glucogenic amino acids are amino acids whose carbon skeleton is degraded to pyruvate or one of the intermediates of TCA cycle. Then these intermediates form glucose through gluconeogenesis. In the case of ketogenic amino acids, their carbon skeleton is converted either to acetyl-CoA or to acetoacetyl-CoA. Whereas both keto and glucogenic amino acids, their carbon skeleton form pyruvate or TCA cycle intermediates in addition to acetyl-CoA. Let's see the mechanism of ammonia toxicity. Ammonia is toxic to the central nervous system. Excess ammonia depletes glutamate and GABA level in brain. So there will be depletion of glutamate level. To compensate for this glutamate level, alpha-ketoglutarate is used. This utilization of alpha-ketoglutarate reduces the supply of alpha-ketoglutarate to the brain, and its depleted level depresses TCA cycle, which adversely affects the production of ATP. There will be formation of glutamine in the brain cells. This excess glutamine is exchanged with the amino acid tryptophan, which is a precursor of serotonin. This results in hyperexcitation. The symptoms of ammonia intoxication include tremor, slurred speech, blurred vision, coma, and ultimately death. This is ammonia intoxication. So this is about overview of amino acid metabolism. This is today's topic. Thank you for watching.